This wall is coming. So, good morning everyone. How are all of you? All well? Yeah, so am I audible and visible? Audible, visible? In your response? Good afternoon, good morning. Audible? No voice coming. Voice is coming or not coming? Voice, no video, sir. Someone writing. Okay, all right. So, everything is all right. Can we start? Voice coming? Okay. Video is also coming, right? Okay. Everything clear. Very good. So, mitochondria is completed, right now? So, we are starting with plastid. So, start with the plastid. Give your attention. Plastid. So, if we talk about plastid, it is present in plantae and some protist. And what are the some protists where plastids are present? Example is chrysophytes, dinoflagellate, and euglenoid. So these are the some of the protistans other than plant who have plastid. Clear? Later on we are going to classify the plastid into three categories based on pigment present in it. So we are classing the plastid into three categories. The one category I am writing chloroplast, another is chromoplast, and another is leucoplast. So these are the three type of plastids are there. You can say about chloroplast, it is green color plastid. Chromoplast is colored plastid other than green. It is colored, but color is what? Other than green. And if we talk about leucoplast, it is colorless plastid. It is colorless plastid. What are the pigments present in? So here I am writing about color, and then I am writing about the pigments in all these plastids. So in case of chloroplast, pigment is what? Chlorophyll. And chlorophyll mainly present here. And it is a which color pigment? Green color pigment. But only chlorophyll not present in chloroplast, it also contains carotenoid. Carotenoids is there. This carotenoid is of two possible type. One is carotene and another is xanthophyll. So if you talk about carotene, its color is yellow to yellow orange. I mean different shades are there. And xanthophyll color is what? Basically yellow. But who is mainly present? Chlorophyll is mainly present and it is green color, and that is the reason wherever chloroplast is there, that part become green. Got the idea? Now if we talk about chromoplast, here which pigment is there? Carotenoid present. Carotenoids present. Chlorophyll is absent here, carotenoid present. That is the reason that this is not colored, it is other color other than green. Got the idea? Leucoplast, pigment absent. Pigment absent. Got the idea? So that is the reason leucoplast is colorless plastid. Right, so wherever chloroplast is there, what are the function, major function it serve? Function. Wherever chloroplast is there, they perform the function of photosynthesis. Photosynthesis, because chlorophyll is behind that. We will talk whatever who is there. Chromoplast is there. Chromoplast is there. They will serve the function of which one? They will serve the function of, example, present in petal. So what happened? Attract pollinator. Attract pollinator. And wherever, which one? Colorless plastid, sorry, is there, its function is storage. So different plastids serving the different function. So based on what it is storing, on that leucoplast is of 3 type. Leucoplast is of how many type? 3 type. What is storing on the basis of that? Leucoplast is of 3 type. One is amyloplast. These amyloplasts store starch, that is carbohydrate. Another is aleuroplast, and these aleuroplast is also called proteinoplast. So these proteinoplast or aleuroplast store protein, and the last is elaioplast. These elaioplasts store fat or store lipid or oil. Got the idea? So these are the 3 type of storage plastid, that is called leucoplast. Is it clear in the mind? Is it clear in the mind? So this is about the plastid. Getting the things clear in the mind? So for chlorophyll synthesis, light requirement occur. So the part who are not in the light, never chlorophyll is present. So for green pigment formation, always who needed? Light requirement is there. And you can say about pigments are interconvertible even. So I am telling chloroplast may convert into which one? Leucoplast. And may convert into which one? Chromoplast. Leucoplast may convert into which one? Chloroplast. And leucoplast may convert into which one? Chromoplast. So chloroplast convert into leucoplast in absence of light. Leucoplast convert into chloroplast plus light. And here you can say minus light. Getting the things or not? Once chromoplast form, it not reverse. So chloroplast to leucoplast is reversible, but once chromoplast form from anyone, it is not reversible. So you have seen when tomato are young, not mature, it is green color. And when it become mature, it become red color. But when it turn into red color, it not return back to green color further. Getting the idea now? So these are going to interconvergible into each other also. Now we are moving ahead about chloroplast. Now we are going to discuss about chloroplast. So if we talk about chloroplast, where it is present? Present only in green part of plant. It is present only in green part of plant, not in non-green part. Getting the things or not? So an example I am writing where it is present, mesophyll cell. Mesophyll cell. And the cell where chloroplast is there usually perform the function of photosynthesis. So now I am going to talk about where it is present. Occurrence I told it present in green part of plant. Now I am telling about the number of chloroplast. Number of chloroplast. So it is variable in number. It is not like every cell have same number of chloroplast. An example, chlamydomonas. Chlamydomonas have how many chloroplast? One chloroplast. And another example, mesophyll cell. Mesophyll cell have 20 to 40 chloroplast per cell. Got the idea or not? So you are finding the number of chloroplast is variable. Now we are going to talk about size of the chloroplast. So if we talk about the size of the chloroplast, its diameter is 2 to 4 micrometer. And if you talk about length is 5 to 10 micrometer. This is the data of NCERT. Check. It may be data may be wrong. So check NCERT. This is the data given or any data different is given. 2 to 5, 4 and 5 to 10 is given. Is it there? Right? Okay, ji. Now we are going to talk about shape of the chloroplast. Shape of the chloroplast. If we talk about it, have variety of shape. Variety of shape. So if you talk about the shape of the chloroplast, it may be lens shape. It may be discoid. It may be oval shape. It may be ribbon shape. And many more shape. Like in Ulothrix, it is U shape. So many more shape. I am not going to talk about where which shape is there because it is not in boundary of your textbook. Now we are going to talk about a structure of chloroplast. A structure of chloroplast. So before drawing the structure of chloroplast, I just want to write one more line. Like mitochondria, it is also a double membranous semi-autonomous cell organelle. Reason is same. Why semi-autonomous? I told na, in mitochondria because it have protein synthesis machineries. Here also that protein synthesis machinery is there. DNA is there, RNA is there, 70S ribosome is there, protein synthesizing enzymes are there. Able to understand or not? Is it clear in the mind? Is it all right? So this is about the things you must know about also the chloroplast. Here they are showing similarity with which one? Similarity with which one? Mitochondria. It is also double membrane bounded structure, and it is also a semi-autonomous cell organelle. Now I am drawing the structure of a chloroplast. Give your attention. So have a look. So if we talk about the structure of a chloroplast, I am drawing a structure like this. Suppose that this is a oval chloroplast. Got the idea? So it is how many membranes are there? Two membranes are there. It is surrounded by double membrane. So here it is outer membrane, and this one is inner membrane. This one what basically? Inner membrane. Got the idea? Now in inner membrane is not here what? Enfolded like mitochondria. So inside of the inner membrane, fluid filled matrix is there, and it is called stroma. Fluid filled matrix is there, and it is called stroma. Now inside the stroma, many vesicular structure, that is called thylakoid. It is a membranous structure. Get the idea or not? Going to stack over each other and making it is pile up over each other. Like suppose that each one is 1 rupees coin, and I am placing many coin one above the other. So each coin is equivalent to which one? Thylakoid, and they are making a stack. When it pile up, that stack structure who are forming is called granum. So here single one is what? Thylakoid. And these thylakoid like a coin, and many thylakoid stack over each other, and it is called granum. And many granum are there. So granum is a singular word, and its plural word is grana. So more than one stack altogether called grana. Getting the idea or not? Now I am telling two granum joined together with a membranous structure, that is called a stroma lamella. Stroma lamella, or it is also called stroma thylakoid. It is also called stroma thylakoid. Are you getting the idea or not? So if you talk about a single thylakoid, I am giving you an idea, just have a look. If you look a single thylakoid, it look like this. So it is looking like this. This is a structure of a thylakoid I have drawn here. So it is a thylakoid. Thylakoid. If you look the thylakoid, it is called thylakoid membrane. Thylakoid membrane. It is a single membrane or double membrane? Pin your answer. And it is also called frit membrane. Single or double membrane? Pin your answer. I will see your side after writing. So now this structure inside the thylakoid membrane, this structure is what basically lumen of the thylakoid. Lumen of thylakoid. Got the idea? So this is a thylakoid, and many such thylakoid, when stack over each other, they are going to form which one? Granum, and many granum together is called grana, and in a grana, two granum joined together with a stroma lamella or stroma thylakoid. Now, thylakoid membrane contain photosynthetic pigment. Photosynthetic pigment. So if anybody going to ask you a question where the photosynthetic pigments are present in chloroplast, so what the answer you will give? It is present in thylakoid membrane. In photosynthesis chapter, we will discuss in detail about it. Got the idea? Now, answer of this question is what? This thylakoid membrane is very good. Someone have answered it. It is a single membrane. Chloroplast membrane is double membrane. But thylakoid membrane is what? It is single membrane. Got the idea? It is a single membrane. It is not a double membrane. This is a double membrane. Got the idea? So now, inside the stroma, what is present? Inside the stroma, double-stranded circular DNA is present. This DNA is like prokaryotic DNA. Ribosome is there, 70S. It is also like prokaryotic ribosome. Getting the thing? Few RNA is there. Here also. Everywhere where protein synthesis occurs, these things will be present, and it contain enzymes. It contain enzymes, and it contain enzymes for how many work? Two work. First, for protein synthesis, because some of the protein synthesis occur here, and another, it contain enzyme for dark reaction. Dark reaction of photosynthesis. Got the idea? So this is about the structure of a chloroplast. Picture came or not came in the mind? This is the structure of a chloroplast. Detailing about that we will deal in which chapter? Photosynthesis chapter. Is it clear? Is it all right? Now I am going to write about the function of the chloroplast. Give your attention. Function. So first function is photosynthesis. Photosynthesis. And in photosynthesis, you better know it is a long-term batch, so you manage to know it. Two reactions occur. One is light reaction and another is dark reaction. I hope everyone aware about it. Two type of reaction occur in photosynthesis. One is what? Light reaction, another is dark reaction. If you talk about light reaction, this light reaction occur in grana. Where it occur? Grana it occur. And dark reaction occur in stroma. What is light reaction and dark reaction? We will study in detail in which chapter? Photosynthesis. Second function is like mitochondria. It also perform cytoplasmic inheritance. Which inheritance? Cytoplasmic inheritance. It also perform which one? Cytoplasmic inheritance. So which cell organelle is over? Plastid is over. Is it clear in the mind? Plastid is over. Now we are going to discuss about ribosome. Okay, ji. So can we start? It is done. Did you understood? Diagram came in mind? It is itself a double membrane structure. Inside stroma, many thylakoid pile up and making which one? A granum. More than one granum is called grana. Two grana connected with stroma lamella or stroma thylakoid, and beside of that, in a stroma, who is there? What we are finding? DNA is there, 70S ribosome is there. What is there? Enzymes are there. One student asking a question, why we won't see mitochondria without staining? Not only mitochondria, many cell organelles. Actually, what the problem of the NCERT? NCERT wrote for something regarding, and people get confused, means staining only required for which one? Mitochondria. Because stain the mitochondria with a dye that is called vital dye, that is genus green B, and stain it to increase the refractive index, and you can visualize it better. The things. So it is not applicable to mitochondria. We are staining the cell. What happened? NCERT wrote for a particular point, so student get confused. It is only require staining in mitochondria. Clear? Now I am moving ahead. Now we are going to talk about ribosome. Right. So it is, when you see the cell with electron microscope, you will find it is observed as granular structure present inside of cell, and it is observed by, it is a granular structure. When you reveal the structure through electron microscope, it is observed as granular structure present inside the cell. And who observed it first of all? It is observed by George Pallet. And on honor of his name, we also called Pallet particle. Ribosome also called Pallet particle. Because who is the discoverer? Pallet was the discoverer. Got the idea? Now if we talk about, it is a non-membranous cell organelle. It is a non-membranous cell organelle. Right. And it is also called organelle within an organelle. Within an organelle. The reason behind it, mitochondria is a organelle, there ribosome is present. Plastid is a cell, chloroplast is a organelle, there which one is present? Ribosome is present. So this is the cell organelle present within the other cell organelle, and hence it is called organelle within the organelle. Getting the idea or not? Now, if we talk about composition, I am writing, composition of ribosome. So it is made up of two components. One is ribosomal RNA, another is protein. And how many protein? 80 different protein. That line written in NCERT, and this question also came in which exam? NEET exam this year. And 80 different protein, this word written in second year NCERT, in molecular basis of inheritance. So there are two components present in the ribosome. One is what? Ribosomal RNA and protein all together join, making which one? Ribosome. Now we are going to talk about type of ribosome. So if we talk about ribosome, it is of two possible types. One is 70S, another is 80S. You better know where 70 and 80S is present. 70 is present in prokaryote, mitochondria and chloroplast, and 80S present in eukaryote. So if we talk about 70S, it is made up of two subunits. One is a smaller subunit, that is 30S, and another is larger subunit, that 50S. And 80S is also made up of two subunits. The smaller subunit is called 40S and larger subunit is called 60S. Now every time we are saying SS, what S stand for? S stand for Swedberg unit. Swedberg unit, and it is unit of sedimentation coefficient. Sedimentation coefficient means what? Rate of settling down. Indirectly, it is measure of density and size. So indirectly we are measuring density and size of anything very smaller thing with the help of which one? These Swedberg unit. Getting the idea or not? So everywhere we are writing SS. So smaller thing, density and size we are measuring with the help of this unit. That is the Swedberg unit. So 70S, smaller subunit is 30S. It is a smaller subunit. Smaller subunit, and it is which one? Larger subunit. If I told you the composition of a smaller subunit, 30S have 18S rRNA. Which rRNA present in 30S? 18S rRNA is present. And if you talk about 50S, it contains 5S and 23S rRNA. 5S and 23S rRNA present in which one? Larger subunit. Are you getting the idea? Is it clear in the mind or not? Now if we talk about in 80S, 40S contain, 40S have 18S rRNA. 18S rRNA. rRNA means ribosomal RNA. And 60S have 3 types. 5S, 5.8S and 28S ribosomal RNA. Are you getting the idea? So which particular type of ribosomal RNA, which present in which subunit, picture came in your mind or not? Is it clear? Is it clear? Now I am just telling you, suppose that this is a smaller subunit of ribosome and this is a larger subunit of ribosome. Getting the things or not? So two subunits join like this. So I am just also writing association of subunit, two subunit of ribosome depend on concentration of magnesium ion. So who play a crucial role in joining to? Concentration of magnesium ion. Magnesium ion joining, performing a crucial role in joining it. There are so many stories there. Concentration amount is there. When dimer formation occur, when dissociation formation occur, when association occur, but we are not going to talk about because it is not in NCERT. Now I am moving ahead. Now I am writing function of ribosome. Function of ribosome synthesis, protein synthesis. This is the function of ribosome. So this is about ribosome. Who is also over? Ribosome also over. Now we are going to talk about cytoskeleton. What we are going to deal? Cytoskeleton. Cytoskeleton. So cytoskeleton is what basically? It is an filamentous network of proteinaceous structure. Means one thing is clear, this cytoskeleton is made up of protein. And it is forming a network like a structure in the which one? Cell. So it is a filamentous network of proteinaceous structure present inside of cell, present in eukaryotic cell. Present in eukaryotic cell. So NCERT wrote 3 types of cytoskeleton. One type of cytoskeleton is microtubule another is microfilament, and the third one is intermediate filament. Intermediate filament. Got the idea? So microtubule is tubular structure. It is tube-like structure. And this microtubule is made up of tubulin protein. I am not telling much, otherwise a lot of explanation you can give, but no need for your exam. So microtubule is what? Tube-like structure. It is made up of tubulin protein. And two type of tubulin proteins is present in microtubule. One is alpha tubulin and another is beta tubulin. Alpha tubulin and beta tubulin. Now I am telling you about microfilament 1-2 line. Got the idea? So what I am writing about microfilament? It is filamentous structure. A structure made up of actin protein. It is also made up of protein, but name of protein is actin. You heard about actin, myosin in muscles. Getting the idea or not? Now I am telling about intermediate filament. It is made up of, simply I am writing composition, keratin protein. So simply I wrote additional to NCERT, which is not in NCERT, is what? Which protein is there in which cytoskeleton? So microtubule have tubulin protein, microfilament have actin protein, and intermediate filament have keratin protein. And where microtubule is present, I am writing. About microtubule I am writing. It is present, I am writing about which one? Microtubule. Present in cilia, flagella, centriole, basal body, spindle fiber. So these all are made up of which one? Microtubules. Getting the idea or not? So I told where microtubule is present, a basic idea came in your mind. It is present in cilia, it is present in flagella, it is present in centriole, it is present in basal body. Getting the things or not? And also present in which one? A spindle fiber. Now I am writing the function of the cytoskeleton. This year question came in NEET. Function. So three function written in NCERT. One function written is what? Mechanical support. Mechanical support. Another function written is motility. This year NEET question. What is the function of cytoskeleton? This question was there, na? Motility. And third function is what? Shape. Provide shape to the cell. Understanding the idea? So these are the function directly written about which one? Cytoskeleton in NCERT. Getting the idea? Is it clear? So one student asking whole chapter for the exam. Yes, it is whole chapter for the exam. Getting the things? So who is over? Cytoskeleton is over. Function is what? Mechanical support, motility, and shape. Now we are going to talk about cilia and flagella. For your batch tomorrow also a class, it may be 1 or 2, is depend upon up to where we are finishing. So better if we go for 2 class for your batch, we can finish it more comfortably. In your classes, some classes lag na in between due to network issues and due to one day mic problem. So somehow lagging. Okay, so by taking 2 classes, we will finish it tomorrow. But meanwhile, we are starting with which one? Cilia and flagella. So cilia and flagella are such kind of a structure who have a different external structure or even location or function, but they have the same ultrastructure. They have the same ultrastructure. Means cilia and flagella have similarity in their internal structure. They have the same internal structure, but they are different from each other externally. So what is the difference in cilia and flagella? First I am writing. Cilia and flagella. First I am writing some differences. Cilia and flagella. So first of all I am writing with size. Who is smaller in size? Cilia is smaller in size. And who is larger in size? Flagella is larger in size. One difference we have seen. Then I am writing about number. So if you talk about the cilia, it is less in number. Sorry, more in number. It is numerous. More in number. And who is less in number? Flagella is less in number. Got the idea? Now if we talk about cilia and flagella location, so cilia present all over the body surface, present all over the body. But flagella usually at anterior surface. Usually at anterior surface. An example I am showing you. Suppose that this is what? Euglena. So this is cyclostomna, and from here who is arising? Flagella is arising. So here who is arising? Flagella is arising. So it is present on which one? Anterior surface. Understanding the idea or not? But you see the paramecium. I am just drawing a paramecium for you. So look here. It is a sleeper animalcule. Who is there? Cilia is there. Understanding the idea? It is present all over. So this is the another difference between cilia and flagella. Now I am telling some more difference between cilia and flagella. I am writing movement and beating. And beating. So if you talk about in cilia, it is rowing movement is there. Getting the things? So rowing movement is what basically? Two type of things are there in rowing movement. One is called power stroke and another is called recovery stroke. So what is the power stroke? Cilia when move, how they are moving? They are moving like a swimmer. You know swimming? When you swim in the water, one is what? You are cutting the water like this. So it is which stroke? Power stroke. And then next time you are putting your hand like this. It is which stroke? Recovery stroke. Now if we talk about movement in case of flagella is what basically? Undulatory. So undulatory movement what? Take a rope and jerk it. Who will form? A wave formation occur. So it is which type of movement occur in flagella? Undulatory movement. If we talk about it have a coordinated movement, cilia and flagella have which kind of? Coordinated beating is there. And it have two type of condition. One is synchronous and another is metachronous. But here it is what you will find independent. So two flagella when beat, move, they not depend on each other. But who have a very coordinated movement? Cilia have a very coordinated movement. Now I am going to talk about the function of cilia. So it have a variety of function. One function is locomotion. Another function it act as receptor. Another function have role in ingestion, have role in capturing of food. Paramecium capture the food with the help of cilia, capturing of food. In your taste bud, suppose that it is your taste bud and it is what? Cilia. And here it is nerves. So this cilia is acting as a receptor for the taste. Either you are eating sweet, so sweet receptor, you are eating which one? Chili, so pain receptor getting induced, getting the things or not. So, but function of the flagella is only one, that is locomotion. Function of the flagella is only one, that is what? Locomotions. Got the idea? So this is about the which one? A structure of, difference between cilia and flagella. Are you able to understand some differences or not? Difference in cilia and flagella. Is it clear in the mind? Now I am just telling you about the ultrastructure of cilia and flagella. I think it will not finish, but as it finishes, it is okay. So ultra a structure of cilia and flagella, one thing you keep in mind, are same. No any difference in the internal or ultrastructure of the cilia and flagella. So if you look at the ultrastructure of cilia and flagella, after cutting its section, you will find two components it have. One is periplasmic membrane. One is which one? Periplasmic membrane is there in the ultrastructure, and another thing is in the ultrastructure of cilia and flagella is called exoneme. Exoneme. So this periplasmic membrane is not other than plasma membrane. You can also say it is plasma membrane. And another component is exoneme. So exoneme have 9+2 arrangement. Exoneme have 9+2 arrangement. What is in this 9+2 arrangement? 9 peripheral doublet is there. Peripheral doublet is there. And 2 is central singlet. Central singlet. Got the idea? So 9 peripheral doublet and 2 central singlet is there. If we draw the structure of a peripheral doublet, it is looking like this. It is one microtubule. With one microtubule, another microtubule also connect. Another microtubule also connect. So this is A microtubule, this is B microtubule, and it is called a doublet. It is called a doublet. Got the idea or not? It is called a doublet. Now, with A microtubule, you will find arms are attached. Who is attached? Arms are attached. Outer and inner arms are attached. So a doublet look like this. Now I am telling about a central singlet. Central singlet structure is like this. It is one C1, and this is another singlet. It is C2. C2 and which one? C1. Make up the same size almost. It is which one? C1 singlet. And it is which one? C2 singlet. So these C1 and C2 singlet join together by which one? Cross bridge. Cross bridge are there. So these are the 2 central singlet, and 9 peripheral doublet are present in which one? Exoneme. How they are arranged in the exoneme, we will discuss in which class? Next class. How they are arranged. 9 peripheral doublet, 2 central singlet, how doublet, 2 doublet connected, what component is there, everything we will discuss about the structure of cilia and flagella in next class. and after that we will talk about centriole, then nucleus. So for your batch, tomorrow is how many classes? 2 classes. Okay, Ji? Is it done? Is it clear? Up to here, any problem? You got a basic idea? Right. Thank you very much. So I'm sharing you the screen. So you can scan it if you wish.